here we are practicing the relationships between the coding strand and the template strand of DNA, and then the relationship between those two strands in the mRNA that's made during transcription, and then also the tRNA anticodons that bring the correct amino acid to the mRNA during ribosomal translation. And so basically what we need to remember is who is complementary to who and who is similar to who. So the coding strand and the template strand are our two strands of DNA that are formed by the double helix. And so we know that those two strands are complementary to each other. So right off the bat on the left here, we can know that if we have a G in the coding strand, that must be a C in the template. Right? We also know that the mRNA is similar to the coding strand with the exception of U versus T. All right, so if we have a U here, that would be a T in the coding strand. And if we have a C here, that's also a C in the coding strand, because the coding strand and the mRNA, again, are basically the same with the exception of U, because RNA has uracil instead of thymosin. Now, the other thing we know is that the mRNA is complementary to the template strand. And so we can kind of take a look at both of these, and so we know there must be an A here because that's complementary to the T in my coding strand and complementary to the U in my mRNA, and there must be a G here, again complementary to both. And my missing part of the mRNA here must be a G because it's similar to the coding and complementary to the template strand. The tRNA brings the correct amino acid to the mRNA, and therefore it has to be complementary to it because it has to bind. So we're back to C, G, A. There's my complementary to the GCU. And we can see from this that the tRNA is similar to the template strand, again because both are complementary to the mRNA. The next thing we need to remember is which strand is read by the genetic code in order to make the template. So when you're using your genetic code, you need to use the mRNA, or alternatively, you could use the coding strand sequence, remembering that T replaces U. But if we look at GCU, and you go to the genetic code, which you guys should all be very familiar with now, if we look at GCU, we will see that that is alanine. And again, I had to use the mRNA sequence or I'm going to get the wrong thing. If I use the tRNA CGA, that's going to give me a completely different amino acid. So when you're figuring out the amino acid sequence, you use the mRNA. And you work it from the 5' to the 3' end. Now the mRNA is 5', 3' again because it's complementary and therefore anti-parallel to the template, and again similar to the coding strand. And this means that our complementary to the mRNA, the anticodon, has to be opposite once again to the mRNA, and so forth. All right, so from this we can start to use those same principles to figure out everything else in this chart. So in the second column, this one's a little bit harder, but we see that we have a leucine there. And so if I look on my genetic code for leucine, I can look around and find leucine, and when I find leucine, I see that basically it has a C, U, and then any of the third nucleotides. It's got degeneracy at the third nucleotide point. So CUU, CUC, CUA, and CUG are all leucine. And since I've given my C here, I know that the first two must be C and U, and again we have to remember that it's from the mRNA that we're reading that genetic code. Now with the C there in my anticodon, I know that this codon for leucine was CUG, and then from there we just work our way through based on complementarity and what's similar. I know that the coding strand is similar, CTG, and I know that the template strand is complementary, GAC, and it should be the same as my anticodon. There we are. Now if we look at GUC, same thing. We've got CAG there for my mRNA. That means that my coding strand is also CAG, and then that means my complementary template strand has to be GTC. Remember it's C because we're in DNA. And then if I look at the mRNA sequence, CAG, that is glutamine. And then with these last letters, I can figure it all out again. So T, because coding's complementary to my template strand, which has A. Then we're over with a C here, again because of complementarity. The mRNA A is complementary to the template strand and similar to the coding strand. And from here I just continue to work my way around based on my knowledge of complementarity and which strands are similar. And then U, remembering U's instead of T's if it's RNA. And if I look at TGA, oops, sorry, that's my mistake. This should be a U, remembering that U's instead of T's for mRNA. UGA is a stop codon. So again, the basic rules that you need to remember to do this are that the coding and template strand are complementary to each other. The template strand is read from 3 to 5, and the mRNA is made by complementary base pairing to the template strand. So the mRNA is complementary to the template, and since the template is complementary to both the coding and the mRNA, that means the mRNA ends up similar to the coding strand. And the tRNA ends up similar to the template strand because it's again complementary to the mRNA. And then the last rule to remember is that you read the mRNA, or you could read the coding, but you read the mRNA in order to get the amino acid sequence.